Oh look, a chromosome! Yes, one of those structures found in the nucleus of your cells that are made up of DNA and proteins. There's another chromosome. Wait, please, you're too close. You might stick to each other and we don't want that. Come on, stop it! Oh yeah, you got telomeres. The ends of chromosomes that prevent chromosomes from fusing. If we uncoil a chromosome, we'll end up with one long DNA molecule and some proteins. At the end of these linear chromosomes, we find the stars of our video, telomeres. These extensions of DNA are found mainly in eukaryotes like us, humans. Most prokaryotes have circular genomes. Their DNA does not contain telomeres. If we look a bit closer at the DNA molecule of a chromosome, we can see that telomeres are quite distinguishable. Not only you find them at the ends of the molecule, but they are sequences of nucleotides that repeat 50 to several hundreds of times. These repeated sequences are made up of 6 to 8 base pairs. We humans have the following sequence in our telomeres: TTAGGG on one DNA strand, which can then pair up with the matching strand of DNA AATCCC. Telomeres can be made of both single and double-stranded DNA. At the very end of the telomere, there is a 300 base pair overhang which can invade the double-stranded portion of the telomere, forming a structure known as a T-loop. You can think of this loop as a knot, or a cap at the end of shoelaces. A-G-L-E-T, don't forget it. This knot is special because it stabilizes the telomere and prevents the telomere ends from being recognized by the machinery responsible for repairing the DNA molecule. Non-homologous end joining is a pathway that repairs double-stranded breaks in DNA. If non-homologous end joining occurs at the ends of telomeres, chromosomes would end up fusing. The T-loop is maintained by several proteins collectively called a shelterin complex. And this complex is known to protect telomeres in many eukaryotes from DNA repair mechanisms, as well as to regulate telomerase activity. Now, telomerase is a very important enzyme that adds DNA to telomeres. Next, in this video, you'll understand why having long telomeres is a good thing. So let's talk about their function. As we mentioned before, telomeres prevent the chromosome from deteriorating and from fusing with chromosomes nearby. Since they are long, repeated sequences of DNA that don't code for anything, they work as disposable buffers, or the shoelaces blocking the ends of chromosomes. A chromosome has approximately 150 million base pairs. If we look at the telomeres of a human white blood cell, you'll see that newborns have 8,000 base pairs. Adults have 3,000 and elderly people 1,500. And as you can see, the older you get, the shorter our telomeres become. But why does that happen? Each time a cell divides, it loses an average of 30 to 200 base pairs from the ends of its telomeres. And cells can divide 50 to 70 times. Their telomeres will eventually shorten to the point where the cell can no longer divide and die. But some cells in our bodies have telomeres with superpowers. I'm talking about the cells of your heart muscle. These cells don't continuously divide and therefore their telomeres won't be shrinking. So, João, are you saying that telomeres are the secret to longevity? Well, scientists continue to study these structures, hoping to find answers about how we could live a long and healthy life. Maybe we'll discover the fountain of youth in telomeres. Who knows? A good way to keep your brain strong and healthy is to learn more. So click on the next video to learn more about the human body. Like the video so you tell me in YouTube you want to see more of these videos. Hit the subscribe button so you don't miss out on the next upload. I'll see you in the next one.